heme catabolism, if we talk about heme catabolism, you need to understand what are the sources of heme. So, most famous source you already know is hemoglobin. This is the predominant source. Okay, predominant source. And then we have other sources, other proteins that contain heme. We have done this protein. So cytochrome 450 group of enzyme. Cytochrome 450, why 450? I've told you it is the blue light, because heme inside such protein absorb blue light 450. So we will talk about this blue light also in this class. I will talk. Blue light has significance. So this blue light will be absorbed by heme. That is why this is called cytochrome 450 system and other heme protein. So there are these three categories of protein in our body. One is hemoglobin. Second group is heme containing is cytochrome 450 system. Other are there heme protein. Various heme proteins are there. So these heme proteins are basically, they are enzymes. Many of them are enzymes. Okay. So these three category of heme are degraded, catabolized. This is called heme catabolism. Predominant is this. So what will happen? You know, this cytochrome 450 system is present in liver. Isn't it present in liver? It is present in liver, because liver is very favorite destination for drug metabolism. And this is a very important, hepatocytes are very important tissue for drug metabolism. That is why liver endoplasmic reticulum has abundant amount of cytochrome 450 system. Therefore, liver also has heme catabolism. Your RBC after it, the RBC dies where? Where does RBC dies? Can anybody tell me? What is the graveyard of RBC? Red blood corpuscles. If you don't answer me, I'm going to complain to Dr. Amrita. Sonia, Ketura, Nadia. Anyone, please?
Sir, would it be the spleen?
RBC. Spleen, yes, it is spleen. It is spleen, okay? So, RBC will be degraded in spleen. Here in this, some of this RBC.
Will yes? They also like die in the liver? I mean, like I thought it was.
Yeah, in liver, cytochrome, not all hemoglobin will die. Hemoglobin will not die in liver, it will die in spleen only. But the cytochrome 450 system, the heme that is there inside cytochrome 450 system will be degraded inside liver only. Okay. And other heme protein, generally what happens, bone marrow, your bone marrow has products. So wherever you see production of heme, so heme synthesis, not all heme synthesis will be perfect. Some heme synthesis will be defective. That heme has also to be degraded. That heme also has to be degraded. So this heme will be degraded where? This heme will be degraded in that tissue. Generally, the heme synthesizing tissue is your bone marrow. So bone marrow has mechanism to destroy this heme. Heme is produced because of destruction of RBC in spleen. Spleen has mechanism to destroy this heme, to catabolize heme. Liver has very high amount of cytochrome 450 system. That is why liver has mechanism to destroy this heme, catabolize this heme. Generally, this heme catabolism occurs in this reticuloendothelial cell. So heme catabolism, from heme to bilirubin production happens in reticuloendothelial system. These are reticuloendothelial system, which in reticuloendothelium, it is done by macrophage. Macrophage is also a reticular endothelial cell. Macrophage, it is done inside the macrophage. Okay, inside macrophage, we will see that reaction. Then, so let's first see this reaction of heme synthesis in context of spleen, and then we will utilize elsewhere. So spleen, okay, spleen, heme degradation from heme, there will be production of what? Bilirubin. Okay, bilirubin. And this bilirubin is unconjugated bilirubin. Okay, unconjugated bilirubin will be produced in heme. Now there you see the liver is there. Liver with the spleen, liver is there. It's through splenic vein, and then other supermesenteric vein will fuse. You know, it will form hepatic vein, and through this hepatic vein, this portal vein, this through a splenic vein and this portal vein, it will reach liver, and in liver it will be hepatocytes are there. Hepatocytes will uptake this bilirubin, and then what does it do? It conjugates this bilirubin, and conjugation makes it more soluble. Which molecule will conjugate? The conjugation, the molecule utilized in conjugation is, we have already studied, glucuronic acid, glucuronic acid. So this glucuronic acid, you know, it is synthesized from UDP glucose. UDP glucose converts into UDP glucuronic acid. So this molecule will be utilized for conjugation here. So this conjugation will be, after conjugation, it will be secreted in, you know, it has got bile. So through bile duct, small bile ductules are there at the cell. So have you done histology of liver?
No, sir.
No. So histology of liver, you know, this is portal vein at the center, and there is hexagonal type of thing. Okay, and then there is hepatocyte here, hepatocyte, and these are biliary canaliculi, and then this biliary and vein is also, artery is there. Actually, artery, the hepatic artery also merge into portal vein. So this hepatic artery or this bile canaliculi will join at one place, and it will drain, this bile canaliculi will actually drain the bile into this. Now you can see this is hepatocyte. Hepatocyte at the basal region, there is different type of transporter that will excrete conjugated bilirubin, and bile also. This bile will be excreted from different transport, and this conjugated bilirubin will also be excreted through this bile ductules only, small bile ductules. So this will be uptaken by this here by cell, and then it will transported into bile. It will be taken up by blood, and then it will transported into the bile ductules. So from bile ductules, it will accumulate in bile. So you can see this is secretion step. The secretion step, you can see through bile ductules, the different channel will be there. We will talk about all these channel proteins excreted, and after excretion, it will reach intestine. So in intestine, it will be degraded further. So the name of this bilirubin changes there. The name of bilirubin becomes stercobilirubin. So stercobilirubin in the stool, stercobilirubin. So this is how bilirubin changes. First, unconjugated bilirubin will be formed in spleen, and then through spleen, it will be transported through blood vessel. How does it transported? By binding on the, because unconjugated bilirubin is lipid soluble and it is insoluble in water. It is insoluble in water. Insoluble in your blood. Water. Blood is water. Now what will happen then? This bilirubin will bind with albumin, and albumin will reach here. From albumin, it will uptake. and then conjugation will be done, and after conjugation it will be excreted in the through bile. It will be excreted in your duodenum, second part of duodenum, and then from duodenum it will reach your large intestine. So in large intestine there is colonic bacteria. So colonic bacteria will deconjugate and metabolize still more, and that bile again will be, that actually bilirubin again will be absorbed in blood. And this bilirubin, we will talk about all this process. Okay, so this is called enterohepatic circulation. So we will talk about this in detail. So this is the step, just remember for the timing, these are the steps. You can see these are the steps here. These are the steps. First step is bilirubin synthesis. Second step is bilirubin transport in blood. And after blood, it will reach hepatocyte from all the tissue, from bone marrow, spleen. It will reach hepatocyte. In hepatocyte, there will be uptake, there will be conjugation, there will be secretion, and secretion to bile. Actually, bile sac, what is this called? It will be secreted along with bile, and through bile, it will reach gallbladder. So from gallbladder, it will reach where? In intestine. Intestine bacterial degradation will convert this bilirubin into another form of bilirubin. And that bilirubin will be, some of the bilirubin will be excreted in the stool and some will be reabsorbed. So these are the steps of bilirubin. And remember, if there is, the degradation is happening inside hepatocyte itself, so what will happen? So this bilirubin synthesis will be there inside hepatocyte. So transportation step will not be there because it don't have to go through the blood. Now you can see uptake will not be there, but directly this bilirubin will reach conjugation, and this all process will happen. So understand this context, where happens, how which all bilirubin is transported through blood. So we will, which organ is called graveyard of RBC. So this is spleen. From spleen only you can see through this splenic vein, through portal vein, it will reach, the bilirubin will reach hepatocyte, and in hepatocyte it will be, what will happen? Bilirubin will form, bilirubin through bile duct, it will reach, actually bile duct, it will reach gallbladder. From this gallbladder, it will accumulate in gallbladder. From this bile will intestine, and then from intestine it will be some of the degraded by bacteria, and some will be excreted and absorbed. Remember the whole context of bilirubin catabolism. Now, as I have told you, three groups of heme protein. First is hemoglobin, and third is heme from hemoglobin. Third is cytochrome 450 system, and third is hemoprotein. So you can see here, hemoglobin and myoglobin you can put together. Okay, this hemoglobin, myoglobin. Then you can see cytochrome 450 system that are present here. These are heme protein. These are all other enzymes. These are not actually similar to cytochrome 450. These are other enzymes. You can see nitric oxide synthesis that we discussed. Also belong to, in our pentose phosphate pathway application of NADPH, we studied nitric oxide synthesis. This synthesizes nitric oxide from arginine plus NADPH, DPH, and oxygen. So this is required to synthesize nitric oxide from arginine, and then citrulline is formed. So we have done this. This is also heme protein. Peroxidases, these peroxidases have heme. This is also heme protein. Catalases, these are heme protein. These are heme protein. These are not actually hemoglobin or cytochrome 450 system. These are heme protein. And one more we will talk about today. The enzyme that degrade heme is also heme protein. The enzyme is called heme oxygenase. So this three group of protein will actually cause what? They will cause heme. Actually, they will have heme inside. And the heme will be degraded. Source of heme are these proteins. Main is heme, hemoglobin, myoglobin. Second source is cytochrome 450 system. And And third source is hemoprotein catalase peroxidase nitric oxide synthase. So you can easily now, you can understand how heme is catabolized. Main heme catabolism happens in spleen, because you know, the main source of heme is your hemoglobin. That is why graveyard is spleen. And therefore in spleen, the reticuloendothelial cell of spleen, that is reticuloendothelial macrophage of spleen will degrade 80% of the heme. It will contribute 80% of the bilirubin. Now liver and bone marrow, then followed by liver and bone marrow. So you can see here what will happen. This in you conceive this is spleen, and macrophage. What will happen? This will be degraded here. After hemoglobin degraded in the inside spleen. Actually, it is not, hemoglobin is not, this RBC is not broken down into this heme, this macrophage. Actually, RBC breaks down and hemoglobin, HB, will be released. And this macrophage will engulf this hemoglobin. Okay. And after this engulfing hemoglobin, globin will be degraded by lysosome into amino acid. And this will recycle. And heme produced, the heme will be catalyzed to bilirubin. Okay. Heme will be catalyzed to bilirubin. We will talk about this part. And it is not necessary for you to, you already understand how any protein is degraded into amino acid. Protein, similar to protein digestion, there will be proteases that will digest this globin into amino acids in lysosome. And it will be reused. This amino acid will be reused. So this bilirubin will, unconjugated bilirubin, remember, it will reach liver and it will be conjugated in liver. And then what will happen? This bilirubin, conjugated bilirubin, will reach this intestine, large intestine, from a small, large intestine. Now you can see in large intestine, bilirubin will be converted into urobilirubin and stercobilirubin, because bacterial degradation of this bilirubin. So urobilirubin can be absorbed here. Okay. The actually structure of these two are same, because it is appear in feces, that it is called stercobilirubin, and it goes into the blood circulation, and through blood circulation, it will reach urine. And the color of urine is because of this urobilirubin. Okay, because of this urobilin, not because of the bilirubin. It is because of urobilin, degraded product of bilirubin. Okay, so this gives the color to urine, and then this stercobilin gives color to stool. So this is how degradation of heme happens here. And the same way you can see the degradation happens here also, here also. So it is not shown here. Anyway, you can understand this heme is, iron is also recycled and amino acid is also recycled this way. And you know, don't forget this, erythropoietin is released from kidney. It is released from your kidney. Don't forget this, really, erythropoietin is released from kidney. And if the kidney has low oxygen, if low blood supply, or if blood has low hemoglobin, kidney respond to that hypoxia and release erythropoietin, and this erythropoietin will stimulate this synthesis of RBC. Now, so synthesis of bilirubin, we have already done this. Now it is not a big deal. You can understand yourself. So from hemoglobin and hemoprotein, heme is produced. So globin will form amino acid. Now from heme, the two-step reaction that happens inside macrophage. Okay. That happens inside macrophage. First reaction is, first enzyme is heme oxygenase, and second enzyme is heme reductase. This heme oxygenase reaction actually happens in endoplasmic reticulum, and this in cytosol. So understanding where reaction happens is also important for USMLE. Okay, because sometimes they ask question, okay, where does this enzyme present? They will ask, put the picture of this reaction, and they will ask, where does this enzyme present? So this is present in endoplasmic reticulum, and this is present in cytosol. So you can see the byproduct here. You can see the byproduct, you have seen the toxic carbon monoxide. So here byproduct is, one of the byproduct is carbon monoxide. And here heme is also released as byproduct in ferrous form. So two byproduct you can see here. Now, so this is the first step of heme catabolism. This is the second step of heme catabolism. And you can see in heme catabolism, what is required is NADPH that you have studied. I told you any breakdown detoxification requires what? Heme catab. Any detoxification requires what? What does a detoxification requires? NADPH. Here you can see NADPH is requiring in both steps. So let's see this step. Inside the cell, inside the macrophage, consider this as macrophage. Okay, consider this as macrophage. Okay. So see, consider this as reticuloendothelial cell is macrophage. And in this macrophage, you will see first heme. This is actually what? This is microsomal system. You can see this is microsomal system means endoplasmic reticulum. Inside endoplasmic reticulum of macrophage, what will happen? Actually this heme degradation is three-step process. The same enzyme will actually catalyze this heme, actually open the chain of this heme. You can see the chain of this heme is open, and you will notice that there are I'm not going to tell about that, but there is requirement of 3 actually oxygen molecule for degradation of this. You don't need to understand that also. So you can see NADPH also. 2 NADPH required, 3 oxygen molecule is required, and this will break this part of chain and it will elongate this chain. 1, 2, 3, 4. So 1 and 2 chain, A and B, you can consider between A and B, it will break. And after breaking, it will form like this. It will form an elongated chain. And you can notice this. Here, there is 2 propionic acid. On this 3rd and 4th. In the middle, there is 2 propionic acid. So here at the propionic acid, glucuronic acid will bind. That is why I'm showing this. Glucuronic acid will bind. Okay. Here glucuronic acid will bind. So this is biliverdin. This is biliverdin. This is biliverdin. And biliverdin it has to be reduced. Okay. Bilirubin has to be reduced here. And if it's reduced, it will form. So you can see NADPH required for the degradation, breaking the chain. And second, NADH is required in cytosol. So this biliverdin reductase is present in cytosol. Biliverdin reductase is present in cytosol. This is present in endoplasmic reticulum. Remember this very important thing. So after degradation, bilirubin is formed. Now, bilirubin after formed, it will be transported in the blood by binding with the albumin. And then it will be taken up by hepatocyte. In hepatocyte, so bilirubin will be conjugated into glucuronic acid. Here I told you where is conjugation. Conjugation happens here. Here. So there will be 2 glucuronic acid molecule here. There will be 2 glucuronic. This makes it more soluble. This glucuronic acid, because many OH group, glucose has many OH group plus COOH group. That is why the bilirubin diglucuronate will be soluble. This is insoluble in water and soluble in lipid. Insoluble. Now this bilirubin will be excreted in the bile duct, and through bile duct it will reach where? It will reach large intestine. You can see diagrammatic representation of 3 major state processes: uptake, conjugation, and certain proteins in hepatocytes such as ligandin, family of glutathione as transferase Y protein. It is ligandin Y protein binds intracellular bilirubin. Once bilirubin crosses, there is Y protein. Just remember this Y protein will bind this bilirubin, and don't let this bilirubin to permeate to go out of the hepatocyte, and retain this bilirubin, this ligandin on Y protein. They will bind, and then because of this binding, they will be further catabolized into this. After this is catabolized, it will be excreted rapidly into this. Okay. So this is actually bilirubin catabolism and conversion of this bilirubin into detoxification. And remember, I will tell you one thing, one very important thing. When we talk about this bilirubin, bilirubin catabolism is very similar to drug catabolism. Okay, you will see the step. There is two steps. One step is formation of this bilirubin. Okay, the second step is conjugation. The same way you will see the two-step process in drug metabolism. Remember that also. Any detoxification of body has the same type of modality of detoxification. And here you can study this three disorder. A disorder of conjugation problem. If conjugation does not happen, there this conjugation and the enzyme that will do conjugation is called again glucuronyl transferase, UDP glucuronyl transferase. We have studied glucuronyl UGT, UDP glucuronyl transferase. Glucuronic acid is transferred here. So this enzyme does transfer of glue. Now this enzyme deficiency, based on level of deficiency, if it is completely absent, it will cause this disorder. If it is absent but not actually there is actually the activity of enzyme is preserved, and that as much we require is preserved. So it will cause this Gilbert syndrome. So this syndrome are there, two very important, Crigler-Najjar syndrome and Gilbert syndrome. Two syndrome are because of this. We will study about this in detail. So you can see this enzyme after, this is shown that how glucuronic acid is formed. From UDP glucose, glucuronic acid is formed through dehydrogenation, oxidation. So glucuronic acid is added here. One monoglucuronide, and this is UDP glucuronic acid will add it on this bilirubin monoglucuronide. It becomes di. So two-step process, same enzyme will carry out the conjugation of bilirubin into bilirubin diglucuronide. What is the structure of bilirubin diglucuronide? You can see this is the bilirubin. This tetrapyrrole, pyrrole, 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 pyrrole. In between these two, there is propionic acid. 1, 2, 3. Propionic acid is propionyl side chain. With this propionyl side chain, there is this glucose. You can see hydroxyl group here, hydroxyl group here, hydroxyl group. Why I show structure? To make you more confident. Not that I want to put more pressure on you. So I always tell you, it may not be asked in exam, but it is important for your memory, for your understanding. You can see, if you see the structure, you will not know that there are many hydroxyl group here. There are many hydroxyl group here and one carboxylic group here. What makes this, what is there in glucuronic acid that makes this compound more soluble? You will understand now. Many hydroxyl group and then one COO group. This will make here only one COO group. When it is bound, there will be one COO group, but many hydroxyl. This makes this compound very soluble. So this is bilirubin diglucuronide. Now you can see the summary, whole process here, whole process, what will happen. So what is it? This is the summary. Summary, you already know. So two-step process of this heme degradation, two-step process means two enzyme will convert this heme into bilirubin. One enzyme is heme oxygenase and second enzyme is biliverdin reductase. This is present in endoplasmic reticulum and this in cytosol. So easy. Now it is transported in blood by binding with albumin. This is now reach liver, it will be converted into bilirubin diglucuronide. Easy. So this diglucuronide will reach intestine. In intestine, it will be converted by bacteria. It will be converted into urobilinogen, and urobilinogen will convert into urobilin. It will actually be reabsorbed in blood. And it will be excreted through urine, give color to urine, and it will, some part will also be excreted in the actually feces. It will give color to feces. So here also the feces will be colored because of this bilirubin degraded product by bacteria, urobilinogen, stercobilinogen. This becomes stercobilinogen here urobilin. So stercobilin and urobilin here. So remember, the color of stool is because of stercobilin, color of urine is because of urobilin. In normal condition, I'm talking about normal